Hey everybody, so today or with this unit what we're going to be doing is looking more carefully at ABO discrepancies to recognize them and also to understand what we should be doing to resolve them. So I'm just going to get into slideshow. And okay, so basically what I did was I took the more common discrepancies that we discussed in lecture 5 and I placed them on a new PowerPoint. If we were meeting face to face, I would be asking you all to help me fill this in. So unfortunately, try to guess while you're at home watching this presentation, and hopefully it will make sense as we go through it. So let's look at our first one. So we have the results of our forward typing here. We see a 4+ reaction with anti-A, and we see a 1+ reaction with anti-B. So always what you want to do is break your forward and reverse typing down into two distinct components. And first, let's find out, what do we think? Based on forward typing alone, looking at the information that we have from the testing here, what does this patient type as just with the forward typing? And so what we can see here is very strong reaction with anti-A, which means it looks like they have A antigen, and then a weaker reaction with anti-B, which looks like they have the B antigen as well. So if we're just solely looking at forward typing, we're going to say that this person looks like type A, B with forward typing. Bear with me as I write. Okay, so let's now go to the reverse typing. We know that when an individual has both A and B antigens, Landsteiner's rule states that we would expect to have naturally occurring antibodies only to the antigens that we lack. Since they have both A and B antigens, we expect to have two negative reactions with our reverse typing. And so we see that there is no reaction with A1 cells, okay, which is good, that fits. But then when we go to the B cells, we see a 4+ reaction indicating that they have an anti-B antibody, which would not make sense for AB, okay? So if we are just looking at reverse typing alone, we have to think, okay, well, what blood type would have an antibody against B only? And that would be, hopefully you know it, type A, okay? So this is what we call a discrepancy, where the forward typing and the reverse typing don't agree. So when we did our unit on lecture 5, chapter 5, we basically learned that some of the things we should be looking for with discrepancies are missing or weak reactions, okay? So if we look across all of the reactions that we had testing, okay, the one that really is suspicious, we want to say it's this one, okay? We want to say our suspicious reaction is anti-B, okay? And that's simply because it's a weaker reaction. We know that when we're doing our forward typing, we expect to have 3 or 4+ strength of reactions when we're doing that testing. Okay, and so what I always say to myself is, okay, well, if I eliminated this, let's pretend this was really a negative, would then the forward typing and the reverse typing agree? And so if we got rid of this reaction, it was truly a negative, the person would type as A forward and A reverse. So yes, that would make sense, okay? So we have to think about what are some times, or what are some reasons when an individual might have, you know, this extra B antigen showing up. And we talked about something called acquired B, okay? And so acquired B is a type A person who looks like they have acquired a B antigen. Okay, they don't really have the B antigen, but in our testing, it looks like they have acquired a B antigen. And so knowing the patient's history in this case would also be very helpful because, you know, individuals with gastrointestinal diseases can sometimes have a modification of their A antigen, and it can look like they've acquired the B antigen in our testing. So we want to think, well, how can we resolve this and prove, you know, that this person really is type A? There are actually two things we can do. So the first thing we can do is test. This is really hard to write. Test reverse typing with, you know what? I'm going to do something different. I'm going to erase this. Okay. I think it's going to be easier. Okay. And I'm going to get out of slideshow. Okay. So just bear with me for 1 minute. Stop annotating. Okay. Where's my button down here? Okay. I think it might be easier if I do it this way. Okay. So we said we think that this is acquired B. What's our resolution? One thing we can do, let me just, okay. One thing we can do is test patient serum with patient cells. And we call that autologous testing. Okay? So here's why we're going to say to do that. If we take 2 drops of the patient's serum or plasma and add that to the patient's own 3 to 5% red cell solution, they should not auto-agglutinate, okay, because they really do not have an they really don't have a B antigen, okay? So they have anti-B, their anti-B would not agglutinate their own cells, okay? So that's one thing we could do to prove that they really don't have B antigen, and this should be, I'll say, negative, okay? And then the second thing we can do is test with a different anti-B reagent, okay? And so some anti-B reagents are actually formulated to not react with acquired B antigen. So that's something second that we could do, okay? So just want to go back to this autologous testing because that would be like reverse testing, and review that one more time. So we know the patient has an anti-B, and we expect that in a type A person. However, we really don't think they have this B antigen. We think this is like a fake antigen that's not really there, okay? So if we take our patient's serum or plasma that has their own anti-B in it, and it doesn't react with their own red cells, it's proving basically that they do not have the B antigen. Okay, so that's what that is getting at. All right, so let's move on to our next one. Okay, so here, once again, we're going to look at our forward typing and then our reverse. So once again, when we look at our forward typing, okay, it looks like forward typing, A antigen is being picked up here with anti-A, and B antigen is being picked up here with anti-B. Okay, so, oops. So it looks like forward typing, once again, they look like they're type AB. Okay, when we look at our reverse typing, we see that they are reacting with A1 cells, which means they have an anti-A, okay, and they are not reacting with the B cells. Okay, so this reverse typing, they basically look like they are type B, okay, because someone who is type B would have a strong anti-A, but remember Landsteiner's rule would not have an anti-B. So once again, this is a discrepancy. What is our suspicious reaction? Once again, we want to look to weak or missing reactions. And I'm going to pick my pen And if we're looking across here, this 1 plus is suspicious. Okay, so our excuse me, suspicious reaction is going to be the anti-A. Okay, what do we think is the cause? Well, this is kind of similar to the acquired B that we talked about in the last discrepancy. And so basically there's something called the BA phenotype. Okay, so we're going to call this B, and then they write it like this, A phenotype. But this is very similar to the last one, except in this instance we have an individual who's really type B, who looks like they've acquired an A antigen. Okay, and so what can we do to resolve this? What we want to think is, if we can eliminate this reaction with the anti-A, if this turned into a negative result, you want to say, would then the forward and the reverse typing agree? And they would, okay, because if we could eliminate this to a negative result, then they would forward type as B and they would reverse type as B also. Okay, so the resolution in this case is simply going to be to try a different anti-A in this case. Okay, and so by trying a different anti-A reagent, similar to the last case where we tried a different anti-B reagent as one of our solutions, some manufacturers have anti-A that won't react with this BA phenotype. Okay, so they really don't have A antigen, it's just looking like a weak reaction when we do our testing. Okay, and then that's going to resolve everything, and they will truly be type B both ways. Okay, let me erase. It's really strange with the drawing, because you're drawing from the previous slide carries through to the next one, so I have to remember to erase them every time. Okay. So with our discrepancy number 3, let's look at our forward typing. We look at our forward typing and we see negative reactions with both anti-A and anti-B. So hopefully in your mind you're saying it's type. Anybody? I'm waiting for you to answer and you can't? Oh, okay. We know type O lacks both A and B antigens. So Landsteiner's rule states that when you are truly type O, you should have anti-A and anti-B that are being picked up, okay, on your reverse typing. And so when we look at the reverse typing, we see a very strong reaction with the B cells, meaning there is an anti-B there. But the anti-A is missing. And so this reverse typing, the person actually looks like type A. Okay, so this is a mismatch. So when we look at our reactions, this is not a case of a weak reaction like the previous two we looked at. This one, when we look, we see negative, 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 and then we see a strong reaction here. So we want to believe that this strong reaction with the B cells is truly a real reaction. So this is actually an instance of a missing reaction, okay? And so when we see this reverse typing as type A, we want to think, well, what are we missing here in the forward reaction? What do we need to happen to make this truly a type A? And so, if you're thinking along, hopefully you are, basically this is the missing reaction, okay? So in this case, the suspicious reaction is the anti-A, okay? And we would expect to have a strong reaction with anti-A in someone who is a type A, and we're not getting it, so that's missing. So we want to think, well, what could possibly be causing this, or what could we do to help resolve this? And there are actually a couple of things we could do. So one of the reasons if we think, well, why might somebody's antigen be weaker and not reacting with the anti-A? And this is actually a pretty rare thing, but this could actually be one of those rare subgroups of A that we talked about in class, like A, int, A, X, and etc. Okay, so that could be why they're not reacting here with our anti-A reagent. So as far as resolution, once again, there are a couple of things that we can do. And so one of the things we could do is incubate. Okay, so instead of, adding our anti-sera and our cells and centrifuging immediately like we do, we could add, the anti-sera to the forward typing and let them incubate at room temperature, maybe for 15 minutes, and then take the tubes and centrifuge them. Okay, and then possibly, we're going to maybe see some results. So actually, I forgot I have another slide here. So let me clear all my drawings. Okay, so this is an example of some additional testing we could do. Okay, so what we could do is extend incubation time 15 minutes at room temperature with our anti-sera. Okay, we could also test with an additional reagent called anti-A, B. And so anti-A, B actually is a reagent that we get from group O individuals. And so it's a combo antibody, and sometimes it's able to detect that A antigen more easily than just anti-A by itself. Okay, so this would be tested in the same way that we would do forward typing. We would take a drop of our anti-AB in a new tube, add a drop of our patient's 3 to 5% red blood cells. We may want to incubate for 15 minutes at room temperature, centrifuge the tube, and read. And so we see here that this is actually what they did in this case, and now they're getting a 1+ reaction here. And so this is absolutely then going to confirm to us that this is one of those rare subgroups of A. So to do further confirmation on this, they can actually do molecular testing to determine which rare subgroup it is. But just remember that with these rare subgroups of A, and they're very, very rare, we basically, I think it's only, I forget, but they're very, very rare. But anyway, maybe 1 to 8% of the population, it's extremely rare. And some of them are rarer than others. But basically, they would still be considered a type A. We would not want to call them a type O, because we learned in lecture that, mistyping them as O, they could be given to someone who's type O, and, the anti-A in the type O person recipient could react with this A antigen and cause a transfusion reaction. So we do want to consider that they're still type A, but they're one of the rare subgroups. And most often this would be picked up at the blood donor center when they're screening donors. Okay, they're very uncommon. Okay, but just an example. Okay, so let's move on to number 4. So here, let's look at our forward type. So we see a negative reaction with A, anti-A, and we see 2+. Remember, MF is mixed field with the anti-B. So it's looking like this person has a B antigen, but it's not completely agglutinated. There are some cells that are in solution in the tube. So forward typing is still going to look like B. Okay, when we look at reverse typing, we see they have a strong anti-A that's reacting with the A1 cells and no anti-B reacting with B cells. So that fits. So this is an example where the forward and the reverse typing do agree, but we want to try to figure out why this mixed field is going on. So our suspicious reaction is anti-B. And suspected cause, we would want to get some history on this patient. So the resolution would be to get patient history because possibly what this is telling us is there are most likely two populations of red cells in this person. And so if they recently had a transfusion of, let's say, O RBCs, that would absolutely explain it. Okay, so let's just talk about what's going on in the tube. So this recipient of the transfusion truly is type B. So their B cells in the tube are reacting with the anti-B. However, if they were transfused with type O red cells, we know O red cells lack all antigens for ABO, and so the O cells are remaining in solution in the tube, and that explains the mixed field. So getting the patient history would really resolve, and it's really truly not a discrepancy, but it would explain the mixed field. Okay, we're going to move on to number 5. So once again, we're going to look at our forward typing. This one's very clear cut. We have very strong reaction with anti-A and no reaction with anti-B. So what do you guys think? Type A. Okay. So Landsteiner's rule says somebody who's type A should have anti-B. So we should see a strong reaction only with the B cells. We see a strong reaction with the B cells. However, we also see this weaker reaction with the A1 cells. So it looks like they have an anti-A1. Okay? So what blood group would have an anti-A1 and an anti-B? Type O. So this is a discrepancy. So remember our rules of looking for weak or missing reactions. In this case, the reaction that is the weakest is here. Okay? And so we are going to suspect a problem with these A1 cells. So A1 cells are the suspicious reaction. Okay? So let's think about what could cause this. We have someone who looks like they're type A very strongly. However, it looks like they have an antibody against A1 cells. And so what we want to suspect in this case is maybe this is someone who is type A2. Okay? And so we have to remember that with individuals who are type A, about 80% of the population is type A1, but then 20% of the population is A2. And so individuals who are type A2 have a slightly different antigen structure. And so it's entirely possible that someone who is type A2 can make an antibody against A1 antigen because their antigen is different. Okay, so the A1 antigen is different than theirs, and they might have an antibody against A1. So what is our resolution? There are quite a few things we can do, and I'm basically going to talk about that on the next slide. I actually made a new slide for this one. Let me try to clear my drawings. Okay, and we'll move to the next one. I'll get out of the drawing. My mouse is going, guys. I hate to say it. Okay. So additional testing that we can do on this person. Maybe you recall from lecture that there are these reagents called lectins out there that we can use. And lectins are derived from plants, but they react the same way certain antibodies react when they're combined with red cells. And so there is an anti-A1 lectin. It acts like an antibody specifically against A1 antigen. And you may recall the plant source is Dolichos biflorus, if you like your Latin. So this would be like an additional tube in our forward typing. So we're going to add, you know, a drop of anti-A1 lectin, a drop of our patient's 2 to 5 or 3 to 5% red cells, and we're going to test. Okay. And so here we see we got a negative reaction. Okay. So let's see what happened. The patient red cells did not agglutinate with anti-A1. which means they lack the A1 antigen and are probably type A2. Okay, so that confirms A2. The next thing we're going to do is we're going to grab 3 different lot numbers of A1 reagent A1 cells and 3 different lot numbers of reagent A2 cells. Yes, they make A2 cells as well. And so these 6 tubes are going to be set up similar to reverse typing. So we're going to add 2 drops of patient plasma into each one of these tubes, and then a drop of A1, our first lot, our second lot of A1 in this tube, our third lot of A1 cells in this one, our first lot of A2 cells here, second lot of A2 cells there, third lot of A2 cells in that tube, and we're going to centrifuge for 15 seconds and read. And what we see here is each one of these reagent A1 cells showed 2+ agglutination. Okay, so what does that mean? It's confirming that this patient has an antibody, an anti-A1, that is reacting with the A1 antigen on all of these cells. Okay? When we look at, oops, this is a typo. Sorry. Can change it right now. I'm like, what did I do here? Okay. When we look at our A2 cells, we see no reaction, okay, meaning they don't have an antibody against A2. So basically, this means the patient does not have an antibody against A2 because they are type A2. Okay, so all of this together basically is used to confirm that this person is a type A2 who happens to have an antibody against A1. So we still call them type A. So just remember that when we're looking at A1 and A2 types, they're both still considered type A, and this, this anti-A1 that some of these A2 people have does not impact transfusion. Okay? Okay, let's move on. So we're moving on to number 6. Let's look at our forward type. 4+ with anti-A, 4+ with anti-B, the rarest blood group of all, type AB. Since this person has both A and B antigens, we expect them to lack both antibodies, okay? And so we see they do not have an anti-A. However, we're seeing a weak reaction here, which looks like they have an anti-B. And so this reverse typing, which blood group has anti-B? Type A. Okay, so this is a discrepancy. So forward typing as AB, reverse typing as A. We have to resolve it. So once again, we want to look at our reactions and think, well, which one is missing or weak? And in this case, it's an instance of a weak reaction. So we want to suspect our B cells because they are reacting more weakly. What could be the cause here? This is an interesting one, and so I have another slide where we're going to look at how to resolve this. But basically, we want to think that, and I think I mentioned this to you guys before, when we purchase these A1 and B cells from the reagents from a company, yes, the A1 red cells have A1 antigen. Yes, the B cells have B antigen. However, they come from donors, and so they have other antigens of other systems we're going to learn about later on them as well. And so we want to think that possibly our patient has an antibody that's reacting with some other antigen that just happens to be present on these B cells. Okay, so it's basically an extra antibody of some sort. Okay, so we're going to go to the next slide to resolve this. Okay, so what we want to do is pull out some different reagent red cells that we're going to be talking more about in the future called screening cells. And so we did briefly overview screening cells in lecture 2 and 3 when we were talking about different reagents. And screening cells are reagent red cells that we use to determine if a patient has unexpected antibody. not ABO antibodies, but an antibody to an antigen of another blood group system. Okay, so we want to test our patient with screening cells, and we want to test the patient with their own red cells before we conclude what's going on. Okay, so let's look at the first example. So we took two drops of our patient plasma, we tested it with some screening cells, we got positive reaction. So what this means is our patient has some sort of antibody that is finding its target on the screening cells. And remember, screening cells are always type O because we don't want ABO antibodies to come into play when we're looking for unexpected antibodies. Okay, we see the patient is not agglutinating their own cells. So what we want to suspect here is a cold ALO antibody. It's considered cold because it's reacting at room temperature. Okay, room temperature is considered cold when we compare that to human body temperature. We're going to say it's an ALO antibody. ALO means directed against other because the patient is not agglutinating their own red cells. Okay, so our wrap up of this is the screening cells had a target antigen of this ALO antibody. We don't want to stop there. We would perform a panel to identify what this antibody is. Okay, and so this target antigen happened to be on those B cells that we used in our reverse typing in the last slide as well. If we have a situation where we take 2 drops of our patient plasma, test with screening cells, get a positive, and then also test the patient plasma with the patient's own red cells, and we're still getting a positive, this is telling us that our patient has most likely a cold auto antibody. Okay, so auto means directed against self. Most of these auto antibodies, when a person develops them, react with most cells. So the fact that they're reacting with their own is telling us it's auto, and cold, once again, it's reacting best at room temperature. So, you know, our interpretation here, the screening cells and the patient's own cells had the target antigen of this auto antibody, and that's why we have positive results here. And there are some special techniques that we can do to determine the identity of that antibody, and we're going to talk more about that in upcoming units. So don't worry about that for now. So let's move on to number 7. Okay, once again, we look at our forward typing, 4+ with anti-A, 4+ with anti-B. So our forward typing is looking like AB. We know someone who's AB should not have, they should have negative reactions here, okay? And we're seeing, unfortunately, 2+ reactions with both the A1 cells and the B cells. And so what type basically has both antibodies? Oh, all right, so this is a discrepancy. What are our suspicious reaction or reactions in this case? It's the reverse typing, okay? And I'm saying that because we see weaker reactions here. So these are very strong, and we don't suspect that they're erroneous, but these are weaker, so we're going to suspect reverse typing. Okay, so what could be the cause? We want to think, well, both of these tubes are reacting here. So what could cause that, right? We want to think, and actually a very interesting thing that you can do in the lab when you're doing tube typing, and if we were meeting face to face and we had something like this show up in lab, we would take a drop out of one of these tubes and put it on a slide and look at it under the microscope and see what the red cells are doing to give us a clue. But this might be typical of a patient who has rouleau going on, okay? And so remember, rouleau is caused by excess proteins that are present in the serum or plasma of the patient, coating our reagent red cells and causing them to arrange themselves as stacks of coins. Sometimes those stacks of coins can aggregate together and look like weak agglutination reactions, okay? So what is our resolution? We're going to do a saline replacement, okay? And we're going to review saline replacement on the next slide. We're going to review that technique. Okay, so we talked about saline replacement in lecture 5. So when we have those reverse typing tubes that we're getting those weak reactions in both tubes, okay, what are we going to do? Okay, we're going to incubate our patient's serum and the red cells, and then following incubation, we're going to centrifuge for 1 minute. Okay, so normally we centrifuge for 15 seconds to read a reaction, but in this case, we're going to centrifuge longer because we want to have the cells that are present in the tube settle down, because basically after that 1 minute, we're going to take a transfer pipette and remove the serum from each one of those tubes and discard it. Okay, so this first step is important because allowing the serum and the red cells to incubate first is giving any antibody that's directed against a target antigen on the red cells time to bind. However, we're going to remove the serum and we're going to replace it with saline. So we're going to place it with an equal amount of saline in each tube, give it a shake up. Now we're going to centrifuge for 15 seconds and resuspend our buttons like we normally would to read. So if after we've replaced with saline, we see no agglutination in the tube, we know that that 2+ agglutination we saw on our previous slide was false and was due to rouleau. Okay? So if we go back for a brief minute here, after saline replacement, both of these are probably going to turn to negative. And if they both turn to negative, then the reverse typing is going to reverse type as an AB as well. Okay? Getting back to the saline replacement, okay? If, however, after doing the saline replacement, we see agglutination and it looks like true agglutination in the tube, we're going to call that a positive. Okay? So basically, the saline replacement eliminates misinterpreting results due to rouleau. Okay, so we're going to move on next to number 8, and I actually have an 8A and an 8B because I thought it would be helpful with this one if I gave you 2 examples. So we're going to look at our forward typing. No reaction with anti-A and no reaction with anti-B. Lacking both antigens, we know it's type O. All right. We look at reverse typing. Oh no, we see negative with A1 and negative with B. Type O should have both anti-A that's reacting here and an anti-B that's reacting here. So this is a discrepancy. The reverse typing looks like this patient has no antibodies, and so they are reverse typing as AB. So that's a discrepancy. How do we know what's our suspicious reaction in this case? You know, it's kind of hard because we have nothing. So we want to think that there is some reactions that are missing in this case. And so usually antigen reactions are strong and it's hard to have missing antigen reactions. So we want to think that, remember, in reverse typing, we're using patient serum or plasma to see which antibodies our patient has. We want to see if there are some reactions missing here. And so if there truly are, you know, if these turn positive, then the forward typing, you know, would agree with the reverse typing. So what do we suspect here? Low antibody production. And so basically we want to check the patient's age. So we know in the very young and the very old that they don't produce maybe antibodies as strongly as people of other age groups. So, you know, checking on the patient's age could be, you know, a very good clue here. How are we going to resolve it? We're going to incubate tubes. Okay. And so coming up on the next slide, I think I have, yes, I have an example of what we can do. Okay. So firstly, what we can do is incubate our reverse typing tubes at room temperature for 15 minutes before centrifuging to read the reactions. And that may be enough for us to, let me go back, come up with maybe even 1+ reactions here. And if we did, get 1+ with A1 and 1+ with B, it's going to then type as O, okay? If, however, that doesn't work, there's a second thing we can do, okay? So if, incubating at room temperature doesn't bring out these antibodies, we could take the tubes and incubate them at 4 degrees. It's actually basically taking the tubes and putting them in the refrigerator for 5 minutes and then centrifuging them to read reactions, okay? And so basically, okay, what we see here, if now we're getting, after 4 degrees C incubation, we're getting, a positive reaction with our A1 red cells, positive reaction with our B red cells, and nothing with the patient's own red cells, it's going to confirm that this patient truly is a group O patient. If, however, we get reactions with A1, B cells, and their own cells, it could mean they have a cold autoantibody. So remember, whenever a patient is reacting with self, it means they are agglutinating their own red cells. So, that could pop up as well. Okay, so I have to mention it. So I want to do just one more example with you guys. Let's look at this one. When we look at the forward typing here, we see a strong reaction with anti-A, nothing with B. So our forward type looks like, the drum roll, A. We know an individual who's type A should have an anti-B that's reacting with the B cells, and we see that that's lacking, okay? So this patient has no antibodies, so they're actually reverse typing as AB, because someone who's AB would have negatives here. So this is a discrepancy. What is our suspicious reaction? Okay, it's the B cells that we would expect to react with this person's anti-B, okay? Suspected cause, once again, this could be due to age, okay? And our resolution is going to be to take these tubes and let them sit at room temperature for 15 minutes and then centrifuge them and read them again. And possibly, then if we get a 1+ reaction show up here with our B cells, the reverse typing is going to type as an A as well, because we're going to detect that anti-B that's missing. Okay, so same resolution basically, but I wanted to give you a different example with a different type. Okay, so please review these discrepancies. It is important to understand them. It's really important that you could figure out forward type by itself and reverse type by itself and see when they're different. And then, as we did today, you want to look for some clues. Is there a missing reaction like we had in this case? Is there a weak reaction as we've had in other cases? And so oftentimes they're going to point the way to what's going on. So good luck with this, and we will stop for now.